This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode number 367, recorded on December 11th, 2015. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today here in the TWiV studio, Dickson Despommier.
Otherwise known as the heavy breather. Yes, I'm here. And hello, Vincent.
I never said that.
No, but I did. I'll willingly take that title.
I haven't seen you in a while.
Well, I've been traveling.
Good. Do some more.
Some more what?
Traveling.
Oh, I thought you meant breathing.
It is 15 Celsius.
It's ridiculous outside.
It's cloudy out. It is not winter for sure.
It's the flowers are starting to bloom again.
Yeah, that's right. Let's see. So many people today. Who's going to be next? Joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
How have you been?
Been fine.
Actually, It's been 2 weeks, right?
It has been 2 weeks. And we have great weather here. It's just warm, blue sky, partly blue sky. It's very nice.
You had some snow a while ago, right?
We had snow the third weekend of November, about 10 inches, and was it.
10 inches.
Yeah.
Holy cow.
Yeah.
Also joining us from North Central Florida, Rich Condit.
Howdy, everybody.
How are you?
I'm dandy.
You're home,
Really
right?
good. I am home until Monday.
He thinks it's funny. He thinks it's funny.
Well, no, it's delightful. I'm just laughing because it's delightful. It's 75 degrees, clear blue skies. It's just absolutely gorgeous. So that's all good.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. So it's pretty nice here. The clouds are clearing out. We had a lot of fog this morning, but now it's 57 Fahrenheit, few clouds in the sky, southerly wind. Again, definitely not winter.
And we have one more state to add to the mix. Joining us from Pittsburgh, Pennsylvania, Carolyn Coyne.
Hello, thanks for having me. It is warm here as well. I guess I have to do the weather report. It is also unseasonably warm here, about 60 degrees, partly sunny, a little gray, but not as gray as most winter in Pittsburgh. So,
now, Carolyn, you were on TWiV previously.
I was.
I don't remember the number, but
it was an ASV meeting.
Yeah,
right?
it was. It was an ASV.
Yep.
And I remembered, well, because you wore the right shirt to clip a microphone onto.
I don't remember that part of it, but I'm glad that you do.
It was pink.
It was pink. Oh, lovely.
Pink
nice.
button down with a placket on the front, which was perfect for putting a mic. I said, this is perfect. And you said, I know. That's why I wore it.
Yes, of course.
Control of everything, right?
Exactly.
We also have another guest. These are special guests. You like being a special guest, Carolyn?
What's better than being a regular guest, for sure.
Yeah, it did do less work. Our other special guest is also from Pittsburgh, Pennsylvania. And to make matters even more confusing, he is Coyne Drummond.
Thanks for having me.
You're welcome. And I have to start by asking, now you're a PhD student in Carolyn's lab.
Yep.
When you applied to Pitt, did you know she was there?
I think I did know during the application process, and a friend of mine who was also applying to grad school at the time said, you should definitely join the Coyne lab and be Coyne in the Coyne lab. And I said, there's no way I'm going to be Coyne in the Coyne lab. And then
Here we are.
I actually heard Carolyn on your podcast at that ASV recording. And after that, I went and talked to her and ended up joining the lab.
So even after hearing her, you decided to join her lab.
Especially after hearing.
That was TWiV 193, July 29th, 2012, 3 years ago. Can you believe that? 3.5 years ago.
I know, time flies.
I'll say.
And I was talking about the placenta. I remember that. So
you were.
I do remember the topic.
True.
Oh, yeah. That was pretty cool. Today, you're not going to talk about placentas, though.
Not at all.
You'll talk about something else. But before we talk about that, I want to tell you that this episode of TWiV is sponsored by ASM Microbe 2016. This is a brand new combined meeting of the ASM General Meeting and ICAC, which used to be separate. They're now brought together in a meeting called ASM Microbe 2016. It's going to be in Boston, Massachusetts, and that's June 16th through 20th. It will feature the best microbial sciences in the world and maybe even off-world. Who knows? The meeting will explore the full scope of microbiology and provide a distinct forum for cross-disciplinary study, both in the classroom and in the integrated exhibit and poster hall. Frankly, ASM, I don't know what that sentence means. There's no classrooms at meetings, right? So what are you talking about?
I think classroom is part of cross-disciplinary study.
Yeah, I get that, but I don't know why at a meeting there would be. Anyway,
right.
I should not bite the hand that feeds us, right?
This is
No.
very true.
This is a wonderful meeting, and I criticize in jest.
Yes.
Although I don't ingest, I criticize in jest. This is a unique meeting, and it will spark collaboration and discussion. And in fact, I will be there to do a TWiV and a TWIM. And Helen, you're going to join me for one of those, right?
I should be able to make it out for that, sure.
So if your work touches the microbial sciences, indeed, if you work in the microbial sciences, this is your meeting. Learn, connect, and advance at the inaugural ASM Microbe 2016. You should go to register before January 12th. That's the deadline for abstracts.
That's coming up.
Yeah. asm.org/microbe2016. We thank ASM for their support of TWiV.
And I'm glad they decided to come up with a new name rather than just combining all those acronyms.
Yeah, I'm not a big fan of ASM Microbe, but I should not bite the hand that feeds me.
Well,
right. I mean,
it's the same name as their new journal, so that's annoying. I mean, it's been around for a couple of years, but the journal that used to be ASM News is now called Microbe. Can they not
But
think
if
of
they
a
had
different
combined
word?
the names of the two conferences that they merged, it would have been ICACasm.
Yes,
True
right.
enough.
But it could have been, I mean, microbe, I don't know. It seems like it should be microbial sciences, right, or something. But I
Something,
understand.
yes.
Anyway, I was not consulted. All right, the reason we have special guests today is because of the following. So there's a brand new ASM journal called MSphere, and it is an open access journal, which means that all listeners can go and look at the papers. You don't have to subscribe. And this journal will cover papers in virology, bacteriology, fungi, parasites, and we have podcasts that cover all of these subjects. So when Paul Dupré called me some time ago and said, "Would you like to be an editor?" I said, "No, I want to be an eye editor." And he did it, actually. And so what we're going to do is pick papers and talk about them from that time to time. And then they will put a link in the table of contents. So that's the eye editor's job.
Cool.
So
there's already MBio. What is different about MSphere?
Carolyn, you are an editor of MSphere. Why don't you answer that question?
Three-dimensional.
I had a feeling that this would probably come up. So, the way that we've kind of discussed where MSphere fits into the, kind of realm of, ASM journals, what's different than MBio is, at least the way I looked at it and the way that I think that in my mind is sort of what drew me to even agreeing to be one of the senior editors, is this idea that, good science should be published whether or not it has the sort of red bow on top, as I call it. So that, you can do some good science and have some really good data that tells a story, but maybe lacks a certain, amount of, I don't know, what some may call significance, but that, as I said, I sort of deem more of the red bow on top, right? The hook, if you will. And so at least, the way that I view MSphere, how it fits into sort of, why does ASM need this journal, is that it allows folks that want a broad, range of microbiologists that aren't, let's say, specifically focused, just on viruses. Although why anyone wouldn't want to do that, I don't know. But to sort of give them a really nice journal and a forum to show their really nice science and good science that should be published. And again, be open access so that everyone can access it.
So it's the +1 model.
No. Oh, no. I would say that no.
That's just okay.
Yeah, no, I think that usually, at least with the editors, as we've talked about that, those are not two words we ever like to say in the same sentence as M-sphere. No, I mean, yeah. So I think that we want it to be, again, good science, significant work, but may again lack that perfect kind of big red bow on top, which at least I think is kind of more of what MBio is looking for.
Okay.
So you said the realm word, which means we can use that in the title.
Yes.
Yes, you can use. We've come up with many different titles that you could use based around Coyne.
So I am
a few.
I am an editor of MBio, but this idea didn't come to me. But I have to hand it to Paul Dupre because he said let's do something different, and so he was really on board with this. And you are a traditional editor, Carolyn. Mm-hmm. You'll be receiving papers and handling them, right?
I'm a special guest today, but not a special editor like you. I am just a senior editor at M-sphere. But no, and I will say that's what's really been exciting. And Paul, I think, has been a wonderful sort of force and voice for this, is that, and if you look at the editorial board of M-sphere, you'll see that it's, made up of a lot of mid-level, kind of younger, if you will, people. And I think that was sort of the approach that ASM took, was to sort of tap into kind of what it is that we want at this level, and also going forward, the kinds of things that we want to see. And so, we just had a senior editors meeting last week, and, it's really exciting because everyone's really thinking outside the box of how to make this journal different and how to sort of give it its own identity, but also tap into sort of, the next generation of scientists that are either us or the people that we're training.
So the editors will also recommend papers for us to do on the podcasts, right?
Yes,
so,
absolutely.
A couple of weeks ago on TWiV, we did a paper which was recommended about Candida and coconut oil. Very interesting story. Go check that out. And today we have, it just so happens that it's a paper from your laboratory, Carolyn, and you did not recommend it.
Oh.
I saw it on my own, and I thought we should do it. But in the future, Carolyn, you should keep an eye out for TWiVable papers, okay?
Absolutely.
TWiVable,
absolutely.
a new word.
TWiVable,
this is
absolutely.
Good.
Yep.
And all right, so that is the story, and we hope to regularly feature not only the work, but have people come on, like as you and Coyne are doing today. And M-sphere will send a set of headphones to use to be not broken, but used temporarily, right, Carol?
Exactly. You break it, you buy it, is what ASM told me. So
it's not that expensive, actually.
Still, I don't want to upset ASM.
Yeah, but as you see, it makes the sound much better than just talking into your laptop microphone,
Absolutely.
which many people do. So we're going to actually talk about the paper first, and then if you guys have had enough of TWiV, you can leave.
Never.
Yes. And I know that Coyne will probably need to do experiments. It's Friday.
It's true, yeah.
And you need to set them up for the weekend, right?
Of course.
So let's do your paper. So the paper is entitled A Three-Dimensional Cell Culture Model to Study Enterovirus Infection of Polarized Intestinal Epithelial Cells. So this has been published in M-sphere, that you can go and look at it, everyone, because it's open access, and check it out. And maybe we could start. Let's start, Carolyn. We know your history from the other TWiV, but quick reminder: where are you from?
Originally, or where did I come to Pittsburgh from?
No, originally.
Well, I was born in Michigan. I was a transplant. Oh, here's my phone. A transplant to Florida. So I grew up in Florida. So very close to actually where Rich is currently is where I grew up. And then was a graduate student at UNC Chapel Hill and did my postdoc in Philadelphia with Jeff Bergelson at the University of Pennsylvania before coming to Pitt.
Who at UNC did you work with?
I worked with a guy named Larry Johnson, who was a pulmonologist there. So a lot of my work that I did as a graduate student was sort of centered around the airway.
Okay. And Coyne, where are you from?
So I grew up in Evanston, Illinois, right outside of Chicago. And then for undergrad, I actually moved to Portland, Oregon, and went to Portland State.
That's a good
and so I worked in Ken Stedman's lab as an undergrad there.
Yeah, I was going to say, do you know Ken Stedman?
I do, yeah. I know he's been on TWiV as well.
He has, and I just saw him a couple of weeks ago. I was out there.
He posted some pictures to Facebook, I think.
Yeah, and had breakfast with him before I left, yeah.
Yeah, Ken's great. So I was an undergrad there for a couple of years, and then did a thesis in that lab. And then I was a tech at OHSU for 2 years after that before coming to Pitt.
Portland's a nice place, isn't it?
It's a great place.
Yeah, I liked it a lot. So you've always had an interest in science, or did it just pop up at some point?
Always had an interest. I wasn't sure what field of science really, but then I've been sort of sticking with virology for the last bunch of years, I guess, since about halfway through undergrad.
Best field in the world.
Yeah.
Look, Dickson doesn't disagree. You know, Dickson, parasites have viruses.
I do know that.
So let's maybe start with you, Carolyn. You could explain what is a three-dimensional cell culture model, because I thought I lived in 3 dimensions.
Well, I think there are different ways to define it. So, how we view 3-dimensional cell culture. So we call anything grown in 2 dimensions, anything, cells that are grown on plastic, even things that are, on a transwell insert, some sort of membrane kind of support, we would view as being grown in 3 dimensions. Whereas, anything that's grown in 3 dimensions, of course, as the name suggests, have, 3 different dimensions. But how we grow our cells are on beads. And so therefore, because we're growing cells on beads, inherently they have a 3-dimensional property to them. And so often, and this has been, a really nice base of literature for many, many years, that with many different kinds of cells, and I would say in particular cells of epithelial, derived origin, tend to really respond very well to be grown in these different 3-dimensional culture systems. And so you can do that with things like Matrigel. I'd say the most of what people are aware of in terms of 3-dimensional cell models are based in Matrigel. We're using something, at least in this paper, that's a little bit different than that, which we can certainly, of course, I'm sure we will get into. But again, the idea is to sort of allow cells to grow in a nature that they would grow more in vivo-like.
So you're calling cells growing on in a transwell on a membrane, you're calling that 2D?
Yeah, we call that personally 2D, but I will say that certainly you can find some other papers and even some very nice reviews about 3-dimensional cell culture systems that'll refer to that more as a 3D, but generally we refer to that as 2D. And for us, we view anything in 3D, again, as something that has obviously a more sort of, again, I hate to, but 3-dimensional property to it. So matrigel, enteroids, organoids that people grow, and in our case, we're using beads in 3 dimensions.
So
and
something
what's
in a transwell would be
Matrigel?
sort of 2.5D.
Yeah. No, that's probably true, actually, is that, you could certainly look at it. I would think to look at as a transwell as a little bit of probably an in-between as a,
and what's Matrigel?
It's basically just an extracellular matrix. It's like a solubilized basement membrane that you can use to grow cells inside of. So oftentimes when you take primary cells, and we've actually started dabbling a little bit with primary human enteroids more recently as a follow-up to Coyne's paper to do this, is that it allows kind of a nice thick extracellular matrix for a lot of the primary derived cells to grow in. And because of that, and because they're primary cells, and because this is an ECM substance, they won't grow sort of as a flat surface. They'll often sort of kind of grow, almost on top, in and on top of the matrigel, which is why that usually leads to them being in 3 dimensions.
Right.
So why do you want to do 3-dimensional culture?
Well, for us, it was based upon a lot of the, so the work that I at least had done as a postdoc, which was really focused on this idea of how enteroviruses enter, endocytose enter into polarized cells. One of the big questions that Jeff Bergelson and I, my former mentor, and I always had is this idea of how a virus kind of starts on the top of one of these very complex gut cells in vivo and then migrates over to where its primary receptor is, which is Coxsackie virus adenovirus receptor CAR in the tight junction. And so because of that, you obviously need a model that I think really recapitulates this really differentiated apical surface with microvilli, as well as a really nicely developed tight junction. And so Jeff and I had done this just using a lot of intestinal cell lines and now what I'll call 2-dimensional cultures. And, we got a lot of really interesting information out of that. But the idea was that, when you grow a lot of these lines in different models, three-dimensional cell culture models that recapitulates perhaps the environment they would see in vivo, which is, of course, where they were derived, that they will actually change into cells that then resemble a more in vivo-like phenotype. And so a lot of the questions that I think were unanswered from the work that I had done previously about this, apical surface and tight junction was really difficult to ever test in a regular standard 2D cell culture model, because those cells just don't develop the properties of the gut in vivo that we would need to do those studies. And so that was really what led me, and then of course Coyne, to then sort of look more at this idea of, can we take some of these lines that we've been using and make them more in vivo-like to answer some of these questions?
It's not actually a given that just by growing them on a sphere that they would differentiate or become different types of cells, right?
Not necessarily, no. Absolutely not. And I will say that the really, I think, cool and practical, actually, application or nature of the system that we're using, which I can, kind of go into, but we use something that's called the rotating wall vessel bioreactor, which sounds sort of very fancy. And it actually is fancy because it was developed by NASA. And so therefore, I feel like that makes it fancy. But and so the really nice thing about this system is I think it combines this idea of growing cells in 3 dimensions. And so again, allowing them to sort of grow in any direction they want, but also actually growing them in a system that induces some shear. And so the way that this system works is the cells are grown on these commercially actually available dextran beads that are coated in collagen. They're put inside a rotating vessel. And so within this vessel, the beads, which have the cells attached to them, are constantly in motion. And so not only do the cells grow in 3 dimensions, but they're actually subjected to some shear forces, of course, by the sort of flow of media along their apical surfaces. And I think kind of the combination of those 2 aspects of the system are actually what really drives the differentiation, formation of brush border tight junction that happens at least with gut cells in the system.
So did you ever try to do this
in a slow turning spinner bottle that doesn't accomplish the same thing?
Exactly what I was going to ask. That was my question.
Well, but that wouldn't have been developed by NASA and wouldn't be nearly as cool. No, yeah, I think you can. No, actually, no, because you'd have to sort of, and if you look at, again, sort of not to maybe bring too much to the paper, but figure 1A of our paper has a really nice schematic. And so I think the nice thing about, again, this system is that there's enough kind of volume within what are called, they're called STLVs, which are slow turning lateral vessels, to allow the beads to actually turn in basically a perfect circle. If you set this bioreactor to give an RPM, which, if anyone cares, is usually 19 to 20, you get basically a perfect circle that gets formed by the beads. And that's really what sort of induces the shear. And so you can't really do that nearly as well with a roller bottle, which of course is much more difficult to kind of completely fill with media. The other, I think, advantage of this system is that there's actually a membrane within the vessel, which when I'm at least sort of giving talks, I describe as almost like a placenta, in that it's actually mediating gas exchange. So there's actually gas exchange that occurs at this membrane, and there's a pump in the back of the bioreactor that kind of pumps in the CO2. And again, it's sort of meant to recapitulate, a little bit more what would happen.
And so I would add,
is the membrane lining the walls then?
Or
is it at one end or the other?
It's basically the membrane is right in the very center of that STLV, or the lateral vessel that's turning. And so again, if you look at the schematic, or it's basically right in the center of that vessel.
Yeah, figure
so
one.
The vessel is a pipe, and there's a leaky pipe on the inside releasing CO2.
Exactly.
So as your cells are introduced to the culture, they're non-differentiated, or at least non-polarized, and then as they attach, they polarize.
Absolutely right.
So Coyne, let Coyne talk.
Sorry, I was going to add that the other major difference between this and a roller bottle system is the lack of turbulence. So the vessels are completely filled with liquid, and you need to make sure that there's no air bubbles at all at any time, because that can be really destructive to the cells and the beads.
So, Alan,
so that's different than a spinner as well, because a spinner has a bunch of turbulence.
Exactly.
Yeah.
Good point. Yeah.
So go ahead.
The beads, you're talking about these as 3D cultures, but actually on the surfaces of the beads, the cells are effectively growing in 2D or 2.5D, aren't they?
Yes, exactly. So the cell will form a single layer of the beads, and you can see that in figure 1C. We have a transmission electron micrograph of that. And that's actually, I will say, one of the nice benefits of this system is what we've at least experienced in our transwell-based systems in the past with, lots of different cell lines, is that over time, often they will start to kind of grow on top of each other. And so the nice thing, again, about this system is that as the cells grow on the bead, they'll form, I guess, what you could say kind of more of a two-dimensional, if you will, single layer of cells on top of the bead.
So it's not really so much the three-dimensional. We're going to talk about the differences that these cells develop as they grow in the system, but those differences probably don't stem so much from the three-dimensionality of it as the, for want of a better way of putting it, the absence of gravity.
That's yes, I would say yes. And I base that not only on sort of this paper with gut cells, but we've got some other models that we've developed in the lab that absolutely, it seems to be the actual sort of zero gravity shear induced in the system that induces all of the changes in the cells.
So you clearly need to do your next round of experiments on the space station.
Space,
absolutely.
I know. Coyne is going to go to space. Coyne in space.
Yeah, so
I looked at this video that you posted from Jove, the Journal of what?
Visualized experiments.
Mm-hmm.
Right. And it looks like I just kept thinking about the thousands of different ways that you could contaminate this thing. And saying, Coyne, is this a real hassle?
We've had some bouts of problems with that, and it seems to be an issue. You just need to, they're obviously very thick vessels, thick system, just to make sure that it's autoclaved properly. But aside from that, once you've got the hang of it, it's not too bad.
Okay.
So this whole apparatus, it looks like there are 4 places on the bioreactor in your paper where you could put a rotating vessel. That goes into an incubator, is that right?
Absolutely. And you can actually buy a smaller system. So the company that we buy these from, which is called Synthicon, sells a system where you can just have a single vessel rotating at any time. Mm-hmm. But the 4 vessel that we have fits perfectly inside of an incubator. And in fact, you can usually put a shelf. There's enough space in sort of a regular size incubator to put a shelf on top for your kind of standard cell culture.
And does this need to go in a CO2 incubator?
It does. Yes, it does.
And how does the CO2 get into each chamber?
Well, and that's sort of, had just kind of touched on before, but there's actually a pump in the back of the bioreactor and a membrane within the vessels. And so the pump will actually put CO2, basically pump across that membrane, which is why it makes me think of a placenta, across into the vessel. Okay.
So there's a ton of different cells that live in the gut tract, obviously. And the one that you've chosen is the epithelial cell, correct?
Mm-hmm.
Because I worked on a worm that lived where your viruses go in, and struggled with the concept of trying to get some kind of an in vitro model to work. And actually, someone at Cornell succeeded in making an explant of epithelial cells in the gut and got Trichinella to crawl right into them.
Oh,
so
very nice.
it was quite wonderful,
Dickson,
actually.
Do you think if you added parasites to these?
Depends on the parasite, yeah. Like Toxoplasma would be a great model for looking at
Well,
this.
It's funny you should say that because half of my lab now is working on Toxoplasma.
There you go. And the other one would be Entamoeba histolytica.
All right, so you're going to come on TWiV when that paper comes out.
Absolutely.
Yes. I love Toxo, so I'd be happy to.
Oh, we would love to have you.
You have it?
Do I have Toxo?
We all have Toxo.
Yeah.
Probably are seropositive.
Yeah.
I'm sure I am.
All right, so Coyne, why don't you tell us the kinds of experiments you did? This is your thesis project, I presume, right?
Yeah, that's right. And so I guess one of the important things to point out about the bioreactor is that you can see that we have 4 spots for vessels, but that doesn't mean that that's simply 4 experiments. They're just the equivalent of your T75s or what have you. But once you have finished culturing the whatever your cell line of choice is in the STLV, you can then, it usually takes, we give them 3 weeks in the STLV, changing the media every other day. And then from there, you can take the cell-coated beads out and put them into standard, we usually use 24-well plates, and you can do just your normal assays in that system. So you get quite a few assays out of each vessel.
I see. So you don't do any experiments in the vessels?
We haven't, but we get that question a lot. And it's really just, there's the fact that it's 3 weeks and probably 50 experiments worth of, cells in each vessel. Yeah. And just, I think it'd be a great way to grow up a stock of virus, probably, just given all the cells in there and how well some of the viruses grow in these cells. But
so how many cells are in each of these reactors?
Let's see.
Are you back calculating? I'm trying
to back calculate, yeah.
So when we start the cultures, and again, this is kind of cell type specific. For the Keiko-2 cells, we start with about 6 million cells to 300 milligrams of those cytodex beads, which again are a commercially available bead from Sigma. And at the end of the
30 million?
30 million, yeah.
Okay, so let me understand this. So in your figure 1A, you have the bioreactor, and there are 4 positions, and one of them seems to be being used, right? Mm-hmm. And that, where the label STLV is, that's one chamber, right?
Yes.
It has some ports on the front or something, right? That's how you get things in and out?
Well, go ahead, Coyne. You explain that part.
So yeah, there's those 3 ports that you can sort of make out in that image, and the 2 bottom ones, you basically just attach 5 ml syringes to that point. So that really allows you to get all the air bubbles out just by passing them back and forth between the 2 syringes with the plungers. So that's how you get that turbulence-free environment. And then the bigger plug is where you change the media and add your cells and beads from.
Okay.
So Coyne, how long? I'm sorry. How long do your cells live? Because normal gut cells live about 7 days, right? So you've got it's this explanted culture, and how long can you maintain them in that particular configuration?
I see. You mean once we take them out of the vessel?
Yes.
Okay. I don't know if you want to answer this, Coyne. Go ahead.
We haven't really tested the limits of it, but we've gone up to 3 or 4 days, which is what has been shown previously with other cell lines, taking them out of the system, and they still seem to have their differentiated properties, which I guess we'll get into later.
Right.
All right. So
I had a question because I did watch the Joe video, which I wanted to point out included Andrea Radke, who's a former student in our department here at Michigan.
Very nice. That was a very, very helpful video for us.
Any listeners who want to understand this conversation might want to look at it because it makes sense.
It's pretty hard to grasp just with text or speech alone.
Yes. And they had a term for those shorter things that made me think the whole thing is just all NASA speak. So the STLV, and then the other thing was called the HARV. Was just called the HARV.
High.
Right? Something right out of The Martian is what it sounded like to me. So, yeah.
So sounds like you don't use the HARV.
Yeah.
The HARV, I think, is a different vessel, right? Isn't that right?
Right.
It's
shorter, similar. It's similar. There's less volume in it. And then basically one whole side of it, so the whole inner side of the vessel is the membrane, I think. So it's got a bit better gas exchange to surface area.
Also, if the oxygen tank explodes, you can turn the HARV around and plug it into the STLV.
There you go. You got to
get home. Aries 4 after all.
Yes.
So along the line, 2 questions actually. First, what's the history of Keiko-2 cells? These are an intestinal epithelial cell line, correct?
Yeah. They're a colon cancer cell line.
A colon cancer cell.
A large intestine.
Mm-hmm.
But do you have to induce them to differentiate into epithelial cell-like characteristics, or are there special treatments or anything like that?
Not usually. No, I mean, certainly if you grow these cells, so there's a couple of different clones of Keiko-2 cells. We're using sort of what at least most people refer to as the ATCC clone of Keiko-2 cells. And so, if you just grow these cells on a transwell, also in just a regular sort of plastic dish, or often we'll collagen coat them, after a couple of weeks, they'll differentiate and have certainly some, microvilli, and they'll have tight junctions. And so you don't really need to do anything special to them to kind of get them into at least their enterocyte properties.
Okay, and in the STLV, you grow these for like 21 days, and you seem to have a confluent monolayer of cells on the beads. If you go for another, week, 2 weeks, 3 weeks, are these like contact inhibited, so you don't get any more cells? You arrest at a monolayer on the beads?
I don't know if Coyne, you want to answer that, or I can.
Yeah, I've gone a week longer than that, and you do start to see the cells die off at that point.
Okay. So okay, good. All right.
It probably varies a lot by what cell type you're using.
Right.
But at least
one of the things I like is you can do I assume in the transmission electron micrographs, you're just basically pelleting embedding beads and then doing sections right through the beads.
That's exactly right. Yep.
You get to see. You get to maintain the cell-cell contacts and all the junctions and that kind of stuff, so that you can see it from the side, and I really like that.
Yeah.
All right, Coyne. Now tell us some of the things you did with this.
So using just aliquots of the cells, which you can then count, taking them from the system and putting them into 24-well plates or staining them. So we did a variety of experiments. We did SEM to sort of get a look at the surface, the brush border, TEM, as you just talked about, in order to look at the junctions as well as, again, the border. We did a lot of immunofluorescent staining to kind of look at the different markers of specific intestinal cells to see if differentiation occurred after growing in the system. And then we looked also at the Coxsackie virus receptor and co-receptor, and then we assessed and compared the infection of Coxsackie virus in Keiko2s grown in this system compared to normally two-dimensionally grown Keiko2s.
So in this SEM, the scanning electron micrograph, looks like these beads stick together. Is that right?
They can. So they can form these sort of organoid clusters, as some people call them.
That's what an organoid is, more than one bead. Huh?
Pretty much. In this system, yes. And that's also kind of cell type specific. So we've got a lot of other cells. I mean, when we got the system, we were basically just throwing in any cell type or cell line we had or any of our neighbors had to see how they would do. And, some of the cell lines, a lot of the like microvascular endothelial cells will coat the beads and never fuse between beads. It's sort of cell type specific.
And some of them are pretty picky about media. I know that some of the papers that have been, we originally started out using this media, which we dubbed space medium, because it was developed by NASA, patented by NASA.
Yes, and very difficult to get.
Very difficult to get and to make up.
So if you wanted to talk about the beads as sticking together, you'd have to use a NASA term. They accrete.
Ooh, nice.
So tell us the results of the morphology and the staining. What did you learn from that?
So in figure 1 specifically?
Any of those? I know the first couple of figures you have.
Sure. And so I'll start with figure 1. We looked at markers of differentiation. And so GLUT5 is a fructose transporter that in vivo is localized on the apical surface of enterocytes. And so we wanted to see if that was the localization in our 3D Keiko2s. And so we did immunofluorescent staining, and you can see that from the XZ, the stack that we've got, that the 3D Keiko2 cells have this nice apically localized GLUT5, whereas our 2D monolayers just seem to have basically intracellular, these sort of puncti of GLUT5, so it wasn't properly localized. So that indicated to us that there's a nice change to a more in vivo-like phenotype.
Yeah, that's very cool. You got the 2D and the 3D panels next to each other. You can really see that. That's nice.
Yeah.
You also looked at some Coxsackie virus receptors, right?
Yep. We couldn't leave CAR and DAF out of anything. Yeah.
So are they in the right places? All right, go ahead, Rich.
Absolutely. I don't know if Coyne wants to answer this or
either way.
Yeah. Yes, they are. They're actually they look much better than we normally see in 2 dimensions. Yeah.
So
there's a story about how Coxsackie has to access these receptors and things that go on in the cells. That is, the distribution of the receptors is relevant to that entry story. Could you preview that for us?
Sure. I mean, I guess I can sort of take over that. So the idea that I think makes Coxsackie virus a really, I think fantastic, if not perfect, perhaps model, one could argue, for the entry of a virus into a polarized cell is the fact that it uses 2 different receptors. And it uses DAF, or decay accelerating factor, or CD55, which is a GPI-anchored membrane protein that will go to the apical surface of polarized cells for its attachment. But it requires interaction with CAR to actually undergo uncoding dividing. And so what makes this a great model is this idea that in a polarized cell such as the gut, you have an attachment receptor on the very top apical surface of the cells, but then you have the viral receptor within the junctions of the cell. And so basically, how does that happen? How does the virus go from point A to point B? And that was some of the work.
So I visualize the CAR, the Coxsackie adenovirus receptor. It's in the tight junctions. Is that right?
Yes, correct.
So that's down between the cells, and it's going to be
it's going to be
pretty buried. Right?
Absolutely, completely inaccessible from virus, entering from the lumen of the gut. And that was actually, this idea was really what led me to my postdoc, honestly, with Jeff, was this idea that, why do so many viruses use junction localized receptors? And that was certainly at a time before we even knew what the Hep C entry factors were. And so now this field, of course, is, really exploded. And that was also
this is a common phenomenon with these receptors being located in junctions. Okay.
And it's to me, at least, as kind of a more cell biologist, it's completely counterintuitive, why viruses have sort of evolved to access receptors that are, hidden from where they're entering the cells. And so,
right. So you could think of the virus as a lunar lander coming in, and there are all these mountaintops it could land on, but
yes.
In this case, it's going to stick to a mountaintop and then go down into a deep canyon to get to it.
All right, enough of the NASA, man.
Yes, right.
All right.
So the binding to DAF loosens up the junctions basically, so that they can access CAR, which is what you did as a postdoc.
I did, yeah.
And it is a wonderful model that I teach in my class all the time because it is perfect. It's just perfect.
Great.
So in these cultures, Coyne, is DAF and CAR in the right places?
Yeah, they're properly localized. DAF is, on the apical surface, as you would expect, and CAR is pretty much exclusively in the tight junction.
And on two-dimensional cultures of Caicos, that's not the case, right?
Not quite as much. So we often see in two-dimensional cultures, and we just didn't include it in the manuscript, but because we published it previously. But often you find a fair amount of the DAF is actually more junction localized in two-dimensional cultures.
Okay. Now you also looked at some other markers, Ezrin and occludin. What are the significance of those?
Coyne, do you want to take that? Or
Sure. So occludin is another tight junction protein, whereas Ezrin is a microvilli-associated protein. And so we saw that, again, Ezrin has a nice apical localization, and occludin is pretty much strictly in the junctions.
And occludin is one of those proteins involved in hepatitis C virus entry, right?
And Coxsackie virus entry, correct.
And
entry factor for both, yep.
And Matt Evans over at Mount Sinai was involved in the occludin story.
Absolutely.
Now, this merged, you have different colors, green and red, and the merged is just perfect for the holidays.
Yes.
It's beautiful.
We aim to please.
It is just beautiful.
Thank you.
You also did some transcriptional profiling, Coyne. Why did you do that?
So we wanted to get a sort of idea. Since we saw some evidence of perhaps differentiation and definitely gene localization and expression change by IF, immunofluorescence, we wanted to see sort of on a more global scale what the overall changes in gene regulation were after culturing Caicos in 3D. And so we performed transcriptome, full transcriptome sequencing on Caicos2s that were either grown in normal monolayers or in the STLVs.
Mm-hmm. And then you had to take all that data and make sense out of it, right?
That's true. We had to learn how to do that.
And
we were new
Which
to
was
the
interesting.
process. Yeah.
Well, it's an essential skill, right, going forward.
Yeah.
Mm-hmm.
So it's good that you can learn now so that when you have a lab, you don't have to get someone else to do it for you, as I do.
I had a Coyne to do it for me, so.
Yes.
So what did you find from this? Did you want to ask an essential question, Rich? Of course.
I was going to ask what you just asked.
Yeah, what did you find?
Oh, and so this is figure 4, basically. And we had 2 independent monolayer RNA preps and then 2 of these independent 3D cell cultures. So we got the total RNA from those and then submitted them basically for transcriptomic sequencing. And then we saw that the 2 independent vessels had much more similar levels of gene regulation. And then the 2D monolayers, as expected, had those clustered together when you look at them as in figure 4A. And then, so we performed differential expression analysis using a software package called DSeq2 that you can use in R. And so we basically had a cutoff, a significance cutoff of 0.001, and looked at just the genes that were significantly upregulated or downregulated. And so if you look at the heat map in figure 4b, you can see that the expression profiles really seem to be much more similar and differentially clustered between the 2D cultures and the 3D cultures.
Yeah. R, by the way, was written by pirates.
Yeah, that's good.
Did everybody get that?
Yes, I did.
Wow,
Yeah.
amazing, amazing. Hey, Alan, I made a joke.
It was good.
Not bad.
Good.
Not bad.
So when you look at the genes that are turned on in 3D cultures, do they make sense?
A lot of them do. So a lot of the sort of strongest hits, the most highly upregulated genes in 3D, a lot of them are intestinal specific genes, things like mucins, and then there's an aquaporin, an intestinal peptidase as well.
Mm-hmm.
Speaking of mucins, do you get development of goblet cells or not?
Yeah, so we use, going back to the staining in figure 1, there's a UEA1, which is a mucin binding lectin. So we used a FTSE conjugated one and saw that there was a bit of an increase in apparent mucin production, but it's not really like there's a thick layer of mucus the way that there are with some other intestinal lines like M407s.
Yeah, goblet cells are actually under the control of T cells, by the way. So that's kind of an aside, but maybe throwing in some T cell regulator substances might aid it. Yeah.
You could actually add other cells to this mix, right? And see if they get things that are supposed to be part of the mucosa and see if they get incorporated.
Exactly.
That's sort of the plan. Yeah. And one of the things that Coyne had sort of dabbled in kind of independent of the paper was to even develop, M cell models in the system, which is when,
Nice.
taking enterocytes and growing them with B cells. And so those are things that we're working on, with the enterocytes, but also with some of the other cells that we've been kind of working with in the system.
I would even go so far as to say, as if one of your cultures gets contaminated with bacteria, don't throw it away. Do some experiments.
Right.
So another question is, what is the difference between the CACO2 cells that you have and explants made from small intestine?
Differences in terms of expression differences or morphologic differences?
Or even the ability to infect with the virus?
Well,
which part of the intestine does the virus actually go into?
Well, that's a good question, and I think one that we still don't know. And that was actually one of the reasons that we started using the 3D model, was that I think certainly for Coxsackie virus, there's been a real issue with developing small animal models for the virus.
Sure.
And for a long time, the assumption was that it was due to actually an inability of the virus to bind to DAF. But I think within the last 6 months or so, Jeff Bergelson, my former mentor, published a really nice paper, I think it's in JV, showing that he made actually a human DAF transgenic animal. And our assumption, even when I was a postdoc, was that would then, of course, make a mouse model for Coxsackie virus. And he found that actually wasn't the case, that just having the ability of the virus to bind to DAF wasn't enough. And so some of, I would say, these very basic questions about where these viruses actually bind and infect and replicate in the gut is still unknown for I would say, all enteroviruses.
Vince, how do you feel about your poliovirus?
I feel good about it.
Yeah, you always have.
I feel very good. So this is, we do not know which cells are initially infected by polio. It used to be a big area of controversy when Sabin and David Bodian were working on this, and they did not solve it. And to this day, we do not know. I mean, these cells, as you will see, are infectable by polio, but doesn't mean that they're the ones in situ. Yeah. And I don't know how you could do that, right? Because you can't get
do human experiments.
Yeah, you can't get the human. I mean, in the old days, you could get samples from people with polio, biopsies or something, and look. And that's what Sabin and Bodian did. But you can't do this anymore.
There are no small intestinal epithelial cell tumors to derive your cell cultures from.
Well, we Well, I mean, I'm sure we could use a lot of these lines, of course, are tumor derived, but it doesn't really get to sort of that multicellular phenotype that I think we're aiming for. To answer those questions, you, of course, inherently need a model that has goblet cells, enterocytes, M cells, M-like cells, Panna cells, all the different cells. And that's been a real struggle. And I will say sort of perfect timing, I suppose, wise. I mean, it just even yesterday, Coyne was actually infecting some primary enteroids that we've actually started a collaboration with someone here in our children's hospital to look at human fetal enteroids grown in matrigel, so another kind of three-dimensional system, to maybe begin to answer some of that with human cells, primary cells, at basically what cells are the site of infected, what are the cells being infected.
So if you could humanize
this is probably
a
good time to bring up one of the obvious questions here: why this microsphere-based system? I mean, there are a number of 3D culture systems, and there are even some others that people have developed to try to incorporate multiple cell types into a gut-like structure. What argued for this particular one, or was this the only one that worked?
Oh, no. I mean, for me, at least, when we were sort of, this idea kind of, a few years ago, I was thinking about, I guess, getting back into, I guess, what I referred to as getting back to my roots, which was these questions that had always been kind of hanging over my head and Jeff's head, and we actually still talk about to this day. And, instead of basically using the line of, there's no good models for this, we can't answer them, is to try to start thinking about, okay, there's no good models, let's build a model. And at least what drew me to this system when we were thinking about systems, and there could certainly be others that work as well, if not better, because we haven't tested them all. I will say what appealed to me was, first and foremost, is that I do think what is unique about this system we touched on earlier is this idea of shear and turbulence and these different factors that, you don't get in any of the other systems that use matrigel or any other kind of ECM-based system. But then I will say second to that is, although it seems complex and difficult from the outside, it's really easy to use. The system is really easy to use. It generates, as we sort of alluded to earlier, a tremendous amount of cells. And again, doing even co-culture based models and systems is really easy because, again, if you look at those vessels, there are ports on the outside. So at any time during your culture period, you can take off a vessel, split the vessel into two different components, and in one of the vessels add B cells, for example, and then the other vessel not. And so you kind of have a perfect matched, kind of built-in controls, if you will. So
okay.
I have a question that maybe we've talked around, or maybe we've even said it. Can you use primary cells in this RWV vessel system?
I think in theory, absolutely. Some of it comes down to the cell number that you need to start a culture. So at least for us we've been kind of dabbling with fetal derived gut cells, and certainly you don't get enough primary cells to start a culture with that. We've actually tried primary placental cells in the system. We've been using some actually primary blood-brain barrier cells, and so in theory, if cell number were not an issue, I think you could very easily do these with primary cells.
That's actually what I think some people are trying to get at, that with some of the other 3D culture systems where you'd be able to analyze smaller sets of cells. I'm thinking of the organ on a chip type of approaches.
And so that's a real benefit.
That may be something to approach in an entirely different way, just because you may not ever be able to get the kinds of numbers of primary cells that you can of a tumor line.
Yeah, and the other thing that I guess appealed to me about this system was that it's cell line based, and so again, taking advantage of CRISPR-Cas knockdown, we can obviously then, whereas you can't do of course with human lines or primary cells rather, but then we could actually change these cells, still put them back into the system, and allow them to kind of differentiate into a more in vivo like phenotype.
We can't modify humans and make primary cells from them.
No, wait a second. That was Dr. Coyne, you just stole my idea. I'm dying to ask this question. Okay, So I'm not a virologist, but I am a gut biologist because I obviously studied Racaniello for a long time. So, and that has a gut phase. So what if you took the human receptors for the two receptors for your virus, and using CRISPR, changed a mouse into a human for just those two things, and then did a theory fistula?
Well, go ahead.
I don't know if anybody out there other than you. What is a theory fistula?
Yeah.
It's an exteriorized portion or loop of living intestinal tissue. You can actually work in the gut in vivo, and then you can cordon off the gut tract at various places.
Like a loop, right?
Yeah, it is a loop, and then you open it up, and you can actually observe things in situ.
I will say that the gut models that exist for polio, and also there can be for Coxsackie, the issue actually is more complex than even just the receptor, I suppose, unfortunately, is that in order to get oral infection with polio, at least to significant titers, I will say, and Coxsackie virus, you have to knock out the entire interferon system. And that's certainly true for Coxsackie virus. I think it's also true for polio that you'll get some virus replicating, but not to certainly very high titers if the mice have competent type 1 interferon. So it seems to be a sort of multi-layered problem that may extend well beyond just receptors alone.
Yeah, I keep hearing about humanized mice. I just wonder
how
theory, human are they?
You can take the Despommier genome.
Oh, please, nothing would go
So, Coyne, you finally did some infections. Tell us what you found.
Right. So, of course, the idea all along was to develop a good model for infection. And so for CVB, we went and did some single-step growth curves. And so in Figure 5, you can see that there were infection did occur in the 3D cells. So we looked at viral RNA as well as protein. So there's a Western blot for the major capsid protein VP1. And so you can see that infection does occur along the 72-hour time course there. But it seems that perhaps the levels of both viral RNA and protein are a bit lower at some of the earlier time points. But so we were, of course, excited to see that they were infectable with CVB.
Oh, these Christmas blots are beautiful. I love them.
Very seasonal.
These are Western blots where the bands are fluorescing colors, which, us old guys, we just have, black. But this is so pretty. Is every cell on each bead infected? Do you know?
So that's something we're not quite sure of at this point. Staining has been, for VP1, has been... We're able to see that there is positive staining, but it's not quite perfect. A lot of the staining, the specifications are different than 2D cells. Different fixatives are needed, different reagents and different antibodies work. So it seems like perhaps not every cell is infected, but we're not quite sure yet.
So it's one of the things we'd like to know is whether that tells us anything. So the cells that are not infected, are those the cells that are positive for markers of other things?
Any way to label with fluorescent green protein?
It's Dickson's question.
Are we going to label the cells?
No, the virus.
Oh, the virus. Well, there certainly is a GFB Coxsackie. We don't use it, but it exists.
Then you could see every cell then, right?
Well, that would be the question. Is every cell doing it?
Yeah.
It'd be nice to measure it by flow as well. But.
Yeah.
Right.
Now, did you find differences between the 2 and 3D cultures?
So we went on to actually titer the output virus.
Titrate, dude. It's titrate.
Titrate.
Titer is not Sorry, I'm sorry. Sorry.
Dude, what is this dude?
I'm being friendly.
To titrate by plaque assay. And so what we actually saw was that although the levels of total virus and intracellular virus seem to be equivalent from the output of both the 2D and 3D cell culture systems, the actual amount of released virus was enhanced in the 3D CACO2s.
We don't know what that means, right?
Yeah, and so far we don't have a mechanism for that.
Okay.
Yeah, that's sort of something we're looking at now. We have a few clues, but we're still working it out.
Now, you also looked at these cells by, scanning EM that are infected, and you saw changes, virus-induced changes, right?
Yeah, I mean, we were, when Coyne sort of initially had the result of the 3D cells basically producing, slightly lower but near equivalent levels of viral RNA and equivalent levels of viral protein, this idea that we were getting, a log plus enhancement of released virus, the first thing we were thinking of is, well, perhaps the cells in 3D just die more. This is a lytic virus that gets released lytically, so perhaps it just has something to do with, a mechanism of cell death, timing of cell death, and then that actually goes to figure 6, and so, Coyne did a few experiments to look for that. And there didn't seem to be any differences between 2D and 3D. And so, based upon that, at least we've concluded that, this enhancement of virus release seems to be happening actually non-lytically, because at that time, the cells aren't really demonstrating any significant markers of cell death.
But you didn't see any evidence for enveloped viruses coming out of the cells, right?
It's one of the things that we've actually started really following up on. And so far, the data are still coming in, and, we have like an N of 1 in some cases, but it actually looks like so far that the cells grown in 3D may actually generate a lot more microvesicles, and that perhaps what we might be seeing here is just viruses that have kind of hitched a ride in the vesicles. But again, that's kind of pretty early on. So
let's get it.
So do you process the supernatant from these cultures or something to look for the microvesicles?
Yeah, we just do. There's of course different ways to purify exosomes / microvesicles, but we've just kind of been doing the standard, spin out debris, hard spin, and you can certainly see that the 3D cells, even uninfected, so this seems to occur independent from infection, that these cells just seem to churn out much higher levels of, if you will, microvesicles. But that's again pretty early days. So
you also looked at. You did a transcriptome analysis of infected cells, and you compared 2 and 3D, and you found there were more downregulated and fewer upregulated genes in 3D cells compared to 2D. So what do you make of that?
You want to take that, Coyne?
You can take that one.
Oh, yeah, okay. So, again, we saw this sort of
you
this
could
result.
Flip a coin for this,
There we go. So that's what we thought that the title of the episode would be, something about, 2 sides to a coin or something.
Yeah.
But so, again, we had these results where we saw more released virus that didn't seem to link into lysis. And so the next idea was, well, perhaps they're, genes being either induced or suppressed specifically in the 3D system that, again, is sort of like impacting the ability of these cells. And so I will say, when we got the data, it was somewhat disappointing in that there's not actually a lot of things that change sort of specifically in the CVB infected 3D cells. And so, we see a few things that are downregulated, a few things that are upregulated, but very few. However, I think it's certainly worth noting that, Coxsackie virus and other picornaviruses are kind of powerhouses when they infect a cell, and they shut down, translation, transcription. And so I guess that wasn't a huge surprise to us that we didn't see a ton of genes or transcripts being upregulated. And so we're still trying to actually sort out if any of these sort of transcripts that we identified by RNA-seq actually tell us anything or mean anything, or if they're just sort of, the nature of an infection.
Do any of them stand out as being involved in antiviral responses, for example?
Not that would map specifically for the 3D cultures. So anything that seemed to be more mapping to an antiviral response. So there were a couple of chemokines. We see some NF-kappa B, dependent genes or responsive elements in genes. Didn't seem to map specifically or associate specifically with the 3D cultures. And the things that which is basically panel E and F, were the heat map comparisons of the 2D and E, or the 3D and F comparisons. There, at least for us, wasn't anything that we thought, and we of course went through them one by one for a very long time. There wasn't anything that we felt like, aha, this is the answer. So
right, yeah, there's a tough experiment. Sometimes you would like to take a few of those and check them out by, knock them out, but there's so many and you're not sure, and that's hard. All right, so the last experiment, Coyne, you looked at poliovirus. And what's that
Sure,
about?
yeah. So we did 2 things that are shown in the last figure. And so basically the point of this was just to make sure that the sort of differences that we were seeing with CVB and the 3D Caicos wasn't just an artifact to growing them in a different system. And so for part A of that, we grew HeLa cells in the same system and compared CVB growth, in 2D versus 3D HeLa cells. And so we actually saw no difference in output virus in that system. So that was reassuring that the difference in HeLas, which are of course non-polarized cell lines, and they don't have that restriction of CAR being localized in the junctions. It's localized on the apical surface or the cell surface as well. So we saw no difference.
Did you look at any of those other markers, like in the Caicos cells, with the HeLas to make sure they in fact weren't doing any of that?
No, we didn't look at them in great detail.
Okay.
And so then we, going back to the Caicos, we looked at poliovirus to see how polio grew in the 2D and 3D Caicos. And so we actually saw the opposite effect in this case, whereas the CVB grew better in 3D, polio seemed to have significantly higher output virus in normal 2D cells.
You guys have any ideas about that?
No, we don't. Initially, I wonder how much of this, though, has to do with an entry, which we didn't directly test for with polio, like an entry sort of defect, because, certainly in the 3D cultures, and I don't know that we did the experiment, but I would assume that the receptor is probably, of course, much more buried in the junctions. And for polio, of course, it doesn't have the added advantage that that CVB does, that Coxsackie does, to bind to an apical attachment factor. And so I would sort of assume or suspect that the reason we saw the difference is just largely based upon, the fact that Coxsackie can utilize an attachment factor, whereas for polio, its receptor is going to be buried in 3D. But it might be more than that.
Hey, throw some LPS in there, see what happens.
Yeah. Sure.
That may be, that's what Julie Pfeiffer has found,
exactly right.
But maybe that helps get the virus to where the receptors are. Who knows?
Absolutely. Mm-hmm.
Oh, so Coyne, anything else we should touch on here?
Not that I can think of.
Oh, that's the wrong answer. There's always something. Who is it that what's the matter? You wrote something on a piece.
I thought of another title for this show that you might like.
You have a voice, Dickson.
I do.
Turks and Caicos.
Nice.
They're not Turks. Someone, who is it that, Blossom Domania at the end. Or maybe it was Liana Christea, I said at the end, is there anything else? And she said, oh, there's much more. You'll just have to come back. So that's the answer, Coyne. There's much more. We'll just have to do it another time.
Exactly.
That sounds good.
All right. This is good. So this,
I think my favorite figure in this whole thing.
Yeah.
Well, there's lots of them. The confocal, even the Christmas tree pictures are really cool.
Yeah, they're beautiful.
Figure 3C, the scanning EM of the surface that shows really the difference in the density of microvillus formation in the 2D and the 3D picture. That's a real dramatic demonstration of the effect of the 3D culture on the phenotype of these cells. It's amazing.
Yeah,
I actually have that picture. No, go ahead, Coyne.
I was going to say that was one of the first things that we saw as well, and that was
that would keep you going.
Yeah, that picture's on the wall of my office. I printed it out on a canvas and have it on the wall of my office.
Nice.
Yeah.
Coyne, is this your first paper?
First author, yes.
How long have you been in the lab?
Oh, how long have I been here? 2.5 years.
Yeah, so you got a little time.
Right.
You got a little time left, right?
That's right.
And what's the plan next? You doing a postdoc?
I might be. I haven't fully decided yet. But yeah, I think that's the plan at this point.
Okay, you're not going to medical school, are you?
I'm not doing that, no.
Okay. We just want to make sure,
It's too bad I'm on our admissions committee.
No, we want him to do science, Dickson.
He can do both.
He could do both. He could do an MD, PhD, right?
I would say hire him. If Coyne applies to your lab, listeners, hire him. It's great.
So you might not do a postdoc, but if you did, would you do it in virology?
I feel like every step of the way I've said I'm open to doing something else, but then I do end up sticking with virology, so probably.
Okay. All right. Do you want to stick around or do you want to go?
I think I've got to get going.
Should I stay or should I go? Yeah, that's fine. I understand. You going to do some experiments or you going to go to like TGIF or something?
Experiments first.
All right. Thank you, Coyne.
Thanks again for having me.
Thank you, Coyne.
It was a pleasure.
Great talking to you.
Yeah, good luck.
Thanks a lot.
Yep. Carolyn, you notice I didn't give you the option of leaving.
No, that's fine. I'll try to keep my mouth shut. Although
why would you do that?
It's quite difficult.
No, we enjoy hearing you.
Okay.
Yeah, we have some other things to do here on TWiV. Let's see, what should we do first?
Got a snippet.
We could do. So there's some news that I want to talk about. I got an email from Stephen, who's at Penn, and he wrote, "Dear TWiV team, I'm not sure if this news had reached you, so I thought I would share it. Last week, renowned Johns Hopkins virologist Richard Johnson passed away at 84. As you probably know, Dr. Johnson made important contributions in the areas of neurovirology and human prion disease, including fieldwork in New Guinea. He also trained the next generation of Hopkins neurovirologists, including Janice Clements and Diane Griffin. And he sent a link to the Hopkins obituary. And I wrote a little piece on my blog because I bought his book shortly after I came here to Columbia in 1982. It's called Neurovirology, or Virus Infections of the Nervous System. And I still have it, and it was my Bible for years.
Did he work on Kuru?
Virus infections. Who, him?
Yeah.
I would guess so. New Guinea, right?
Yep.
With Carlton Gajdusek. Yeah.
That's it.
Anyway, so I really enjoyed reading his book, and I got lots of ideas from it. So sad to see you go. And in fact, he had a... Why are you walking on the ceiling, Dickson?
Me?
Yeah.
No,
Did you break out the scotch already,
That's
Vincent?
Jumbo the elephant.
There's no more scotch in my office. That's a thing of the past.
It's too bad, by the way.
I have lost, in an uncharacteristic moment, I have lost my train of thought here. So he actually gave us a figure for the first edition of our virology textbook. So thank you,
Yes.
Richard Johnson, 84. How old are you, Dickson?
Not 84.
All right, so we have 10 years of you left, right?
Maybe.
All right, and I have a follow-up. Somebody named Professor VRR.
Yes.
I think 2 weeks ago, Kathy picked the tardigrade genome sequence as her pick of the week.
Yeah.
And the cool thing about that there was a lot of horizontal gene transfer implied by the genome sequence from bacteria and fungi and plants and archaea, right? I forgot the percentage, but it was really high.
It's around 17%, 16, 17.
So, and then a few days later, a paper was posted to bioRxiv by a different group in
Scotland.
I think it was Scotland, saying that there's very little horizontal gene transfer, and they think there's a lot of
Contamination.
There's a lot of press. Yeah, they think maybe because the Scottish group said they went to great measures to clean the outside of the tardigrades because it's contaminated, and the other group. So bioRxiv.
They also did bioinformatic ways of eliminating bacterial sequences. The Scotland group did.
Yeah, right. And so the other group, which is harder, of course, right? Because the thing is, if it's not integrated and you got surrounding sequences, then you don't know. So that's how you would bioinformatically subtract it. If it's not linked to any tardigrade sequence, you throw it away.
Well, and also on the basis of frequency.
Yeah.
Yeah, that seemed a convincing, compelling argument.
And of course, interestingly, at the bioRxiv site, the other group has commented, "We're looking into it. You know, we're happy that you posted this here for publication." But of course, the press is having a field day, you know, with this,
and will continue for many years to come.
Right. So these two groups, they use different sequencing techniques. The bioRxiv group, I think they used the more conventional, which basically is that you cut up the genome into a whole bunch of little pieces, and you sequence the little pieces, and then you assemble them. The North Carolina group used the PacBio, the Pacific Biosciences sequencer, which is its claim to fame is that it can do really long processive reads. So you don't have as many little pieces; you get longer reads. But in this case, it may have generated some spurious results. It's still up in the air, but this is a very vigorous back and forth trying to figure out what's going on.
I think the point I wanted to make is that I mentioned this on TWiV the other day. In the old days, we had DNA everywhere. It didn't matter because our methods were so insensitive that it didn't matter. But nowadays, high throughput sequencing is very sensitive, so contamination is an issue that you have to deal with. I don't know what the outcome of this will be, but we shall see.
Listen, you get the tardigrade and its microbiome at the same time.
Well,
right. But you have to tell which is which.
This is all true.
Yeah, I mean, the whole key was that it was supposed to be integrated in the genome, horizontal gene transfer, right? So,
where did they get these tardigrades from anyway?
Anywhere.
Yeah, well, that's going to vary.
Both got them from the same source, evidently.
They bought them?
Ordered this is a particular strain of tardigrade that has been characterized, and they both sequence the same species, and so they're pretty well-matched efforts.
Right.
All right.
What I really like about this is this is my first experience really with bioRxiv, which is pretty cool. And I really like the way these two groups are interacting with each other. It's positive. They're saying, oh, yeah, that's interesting. Let's figure this out. It's really good.
Yeah, I agree. It's contrary to how you would have done it, right, Rick?
Oh, yeah, I would have just been griping and...
Well, those pox virologists are notoriously, rival.
Really terrible.
Hey, Carolyn, you know that the pox virologists go golfing together at ASV?
Do they really? Oh, it's a very inclusive club then, right?
Yeah, they go off. They take an afternoon off when everyone else is listening to talks, and they go golfing. And I said to Rich once, I can't imagine the picornavirologists golfing together.
Well, there's a lot more of us, so it'd be harder to get us all in one golf cart, right?
Heck, you have a tournament, it's okay. It's as a matter of
Florida,
fact,
so I know how to golf.
it's not exclusive at the ASV meeting. It is the Poxvirus Open Golf Tournament. Anybody can join us, right?
I don't know.
You don't have to be vaccinated or anything.
I once said, "Where you going, Rich?" And you said, "I'm going golfing. I'll see you later." All right, we have a follow-up from our last episode where we talked about CRISPR, and Steen, who had asked us to talk about it, said, I enjoyed the discussion. Ask and ye shall receive. Another big name in the CRISPR credit discussion is George Church, whose group published on editing back-to-back in Science with Zhang's group. Fortunately, Church does not mind being a bit farther from the limelight. The whole patent battle is silly, he's quoted. There has been much research, and if anybody should be making a fuss about this, I should be making a fuss, but I am not doing that because I don't think it matters. Ooh. I guess he is making a fuss about making a fuss.
Indeed.
And he sends a link to a New Yorker article, and he also has another fun quote, this I really like, from Doudna regarding Jill Banfield convincing her to work on CRISPR. Quote, I remember thinking this is probably the most obscure thing I ever worked on. Further underscores the importance of curiosity-driven fundamental research.
Sounds like a quote from a new Nobel Prize speech.
Yes, of course. This is why you can't tell scientists what they can't work on, because you never know where the discoveries are coming from.
This is true.
I will not pass any opportunity to emphasize that point. I really feel passionately about that. And he also mentions that one of the uses of CRISPR-Cas9 is to target DNA viruses for disease resistance in plants, and gives us links to some papers.
Interesting.
Thank you, Steen. All right, Kathy, can you take Christopher's?
Christopher writes, Hi, TWiV team. Thanks for a great podcast. Keep up the good work. Regarding the new Hepegi virus, could this merely be a recombinant virus that developed following co-infection of a cell with both Hepaci and Pegi viruses? Thanks. And he is from Middlesex University in London. So I hadn't really thought about it.
I think not, because the homology was extended throughout the genome, right?
Yeah.
It's not a matter of it's Pegi here and then goes to Hepaci. It's mixed throughout the whole genome, so I don't think that's the case,
right,
Christopher.
Right. And as I left the office after we did that show, one of the key things was that they had these really great serum samples to use that they got from the NIH.
Right.
And Alan commented on that. And I remembered how when I did my rotation as a graduate student, my first year at the Salk Institute, one of the first things that happened was the health office collected some serum from me and they froze it away. And I suspect that by now they've probably thrown it away. But at the time, it even struck me as being kind of a brilliant, interesting idea.
Yeah, that's a gold mine, right?
Yeah.
How could they throw your serum away, Kathy?
I don't know. And, I doubt if I signed any kind of IRB-like thing, in that
not.
day and age. But,
those are ancient years, Kathy. They would never have any IRBs.
I did the same thing at the Imperial Cancer Research Fund. They bled me when I showed up.
Yeah.
I don't know what they do.
We talked about doing that in Vincent's lab to have students give a serum sample when they first came to the lab, and then track them a year later and see how much their polio titers increase, their polio antibody titers increase.
Well, let's take this one step further. When our first son was born, and after he got his first polio vaccine, I took all his stool samples for 2 weeks.
Oh God!
And I have them in my freezer still.
Oh my God.
Really?
Yeah,
You do not want to eat leftovers at the Racaniello household.
No, they're not at home. They're here in the lab.
Oh, okay.
Dude, I wouldn't do that. Although I have to say...
That's how Phil Miner characterized shedding, right?
I'm just imitating Phil Miner. Although, Alan, I do have to say, when we moved... So I moved from Boston here in 1982. I took all my agar plates with clones and streaks of bacteria on them, and I didn't we got here like at 3 a.m, and so I just went into our apartment and I put them in the fridge. And then they were there for a long time, but there wasn't any food in the fridge or anything. And people would come and look, is there anything to eat here? And they'd see these plates.
Right.
Yeah, you could have some polio clones. Yeah. That's it. Alan, can you take Ye-ons, please?
Yes. Ye-on writes, Dear Drs. Racaniello, Dr. Dove, and Dr. Spindler, I have a master's degree in cancer pharmacology, and I'm a biotech specialist working in a patent law firm in Japan. I write in to follow up TWiV 365. In episode 365, Dr. Racaniello and Dr. Dove discussed the patent issues regarding CRISPR in E. coli. In general, the invention defined by claim language has to be novel and inventive. For novelty, the applicant has to show how the claimed inventions are different from those in the prior arts, such as publication or other patents published before the priority date, in order to overcome the anticipated rejection. The obviousness standard varies from place to place. In Japan, for example, in order to overcome the obviousness rejection, it's generally required to demonstrate an unexpected significant effect in the specification as filed, or there is undue difficulty for those skilled in the art to achieve the claimed inventions. Those who are interested to know more about the Japanese practice can refer to this document, and she provides a link, which is also available on the website of the Japanese Patent Office, pages 29 and 33. Yes, you can tell this is somebody working in a law firm. In regard to this, if the prior art shows one enzyme can be used in E. coli, the arguments simply state that no one has ever used this enzyme in a human cell. The argument simply stating that no one has ever used this enzyme in a human cell is not sufficient. It's required to show why people would not have conceived of applying such an enzyme in human cells. Some applicants publicly disclose their inventions via paper, poster, press release, or the like before filing priority documents, and those publications become priority-destroying documents. So in other words, if you publish before you file the patent, you've just destroyed your own priority. As Dr. Dove mentioned, a patent is not real until it's challenged in court, and it is expensive and time-consuming for applying and maintaining a patent. If the inventor would like to transfer their inventions with commercial interest to a firm, it's necessary to value the compensation to avoid the controversy. In view of the Dr. Nakamura, the winner of the 2014 Nobel Prize for Physics and the inventor of the blue LED, who I think had a patent-related issue around that.
So that's used in Blu-ray, right? Or is that something else?
I think that's different. I think Blu-ray is just the branding of a basic laser. Because that's high definition DVD.
Yeah, you're right.
This could also be used in grow lights for.
Oh yeah.
Yeah.
Right.
Sure.
And Yeon says the weather in the Kansai area of Japan is 7 degrees Celsius, 64% humidity, 0% precipitation, wind 1 meter per second, a chilly night. Thanks for all the wonderful work, and I love TWiV.
Oh, look at this. A patent person in Japan is listening to TWiV.
That's awesome.
That's amazing.
I'm always amazed at who listens. So this is all about Zhang and CRISPR, who, he did it in human cells, but it had already been done in other cells, and so he's got the patent, so maybe that's challengeable. I don't know.
Oh, any patent is challengeable. Yeah. I think this one, I mean, I'm not a lawyer, but just from what I've seen done in the U.S. system, it might be difficult to defend this in terms of obviousness because, this thing has been shown to work this way in E. coli. What's the next experiment that any molecular biologist is going to think to do?
So that was published before the patent filings, that it was working in a certain way in E. coli, right?
Yes, I think so.
I don't know exactly, but that could be an issue with
that could be, and that's something that would be sorted out in the courts.
Okay. All right, thank you, Yeon. Adam writes, Dear comrades of all things viruses, in the most recent episode, the question of DVM versus VMD came up briefly. My former postdoctoral advisor, Lawrence Eisenlohr, VMD, PhD, explained this to me when I was in his lab at Thomas Jefferson University. I asked the same question that you did. What is the difference? Turns out that the vet school at the University of Pennsylvania is the only institution of its kind to award a VMD, which is equivalent to a DVM at other vet schools. I think this came about because the veterinary department was at one time part of UPenn's medical school and then later became a separate school of veterinary medicine.
That's right.
In a similar vein, the Penn School of Dental Medicine awards the DMD to its graduating dentists.
They do.
And they didn't just call them veterinary doctor for obvious reasons.
Right.
So the DMD is opposed to DDS, Dickson?
Yep.
So all their schools give you a DDS. Huh?
Correct.
That's right. I've seen DMD.
Yep.
Right.
I knew someone at that school that got that degree.
So they went to UPenn.
It's funny. My degree from Cornell is called an AB.
Really?
Yeah, I don't know why I don't just call it a BA. I actually call it a BA because I don't like AB. Sounds like other things.
So there.
And one more. Dickson, can you read
Jack
one?
writes. Hi, TWiV overlords. I have done my duty to contribute to the TWiV bump by downloading Vincent's appearance on Perfect Your Podcast.
Perfect.
No, perfect.
It's perfect.
Okay, perfect your perfect. Perfect your podcast. I look forward to finding out.
Nice to Dickson.
That's right. I look forward to finding out how big it was. Your pick of the week was about keyboards. Reminded me of an open source project that is dedicated to bringing steno to the masses. Steno?
Stenography.
Oh, okay. Handwriting, 31 words per minute. Average typist, 40 words per minute. Top QWERTY typist, 120 words per minute. Top Dvorak typist, 140 words per minute. Voice writer, 180 words per minute. Average speech, 200 words per minute. Amateur stenographer, 160 to 200 words per minute. Professional stenographer is 225 to 300 words per minute. And the steno world record was 360 words per minute. The video is worth watching all the way to the end when the presenter does a talk through of how steno can be used for coding. Interesting.
Very interesting.
I had someone in my office, and you knew him, Vince. Is Stephen Chen. He won the high school championship typing contest. He could type 160 words a minute without a mistake. It doesn't say if there are mistakes in here, by the way.
So this open source open steno project,
kind of cool,
which he links to,
is
all about people using steno keyboards, which are different, hooked up to computers so they can type really fast and code really quickly.
Right. And so it's not the stenography that Judy Gaza, the ASV secretary, does. She does the old-fashioned...
By hand, right?
By
writing,
hand.
Yeah. This is with a little machine where you see them...
That's court stenographers.
They're court stenographers. Their hands go up and down. It doesn't look like they're doing anything differently.
That's right.
It's amazing.
It is.
I didn't know they could type so quickly. Now, this speech, average speech, 200 words per minute. Carolyn, you're about 300, I think.
We're talking or typing?
Talking.
Oh, yeah, of course.
Let's see if Rich Condit is back. We can get him to read. No, there's no more emails.
By the way, I'm seeing these people logged into the Google document, Anonymous Frog and Anonymous Orangutan.
Really?
Yeah.
Probably the Coynes.
A frog and an orangutan.
Yeah, the orangutan just came on.
Yes.
Interesting.
Really?
Anonymous orangutan?
Yeah.
I have it up on my screen, so perhaps it is me.
Maybe somebody's aping us.
I want to do a really quick snippet, which is a cool paper published in Biology Direct by Krupovich, Virkate Krupovich, Prangshvili, and Eugene Koonin. And this is a purely bioinformatic paper. It's entitled Evolution of an Archaeovirus Nucleocapsid Protein from the CRISPR-Associated Cas4 Nuclease. So cool. So viruses... One idea as to the origin of viruses is that early in the days of Earth, before cells, there were self-replicating nucleic acids, RNAs first, and then cells arose from them. I just skipped a long time of evolution there. And then viruses acquired a capsid somehow. These nucleic acids acquired capsid genes and became the viruses we know today. And figuring out how this happened is hard because viral capsids don't have any homology with cellular genes, and so it's hard to know where they came from. Now, in this paper, what they're working on in the sequences of a virus of archaea. It's called Thermoproteus tenax virus 1, TTV1, and the archaeon that it grows in is a hyperthermophilic archaeon, which grows at 86 degrees Celsius. These virus particles are filaments 400 by 40 nanometers in size, and they have a double-stranded DNA genome in them. 4 capsid proteins. And this has been known for 30 years, this virus, but these proteins haven't been related to anything. But in this paper, they use a more sensitive homology program called the Hidden Markov model-based HH-PRED analysis. You got that? And they found that one of the capsid proteins, TP1, is homologous to the Cas4 nuclease. Cas4 is one of the nucleases involved with the CRISPR system that's thought to be involved in capture of incoming nucleic acids and chopping them up so that they're then put into the genome for use later on. So this is a capsid protein that seems to have been derived from a part of the CRISPR system. So the CRISPR system is prokaryotic, and this is an archaeal virus. So this capture is going across kingdoms. And
not only is CRISPR prokaryotic, it's antiviral.
It's antiviral. Of course, it's also present in archaea as well, but this particular Cas4 is a prokaryotic one. Now, when they line up the capsid protein with Cas4, the capsid protein is missing the C-terminus of Cas4. It's not the whole protein. And so they went back to the genome sequence of the virus, and they found an open reading frame that nobody had paid attention to, and that's the C-terminus of the protein. So they have an entire Cas4 open reading frame, but it's split. So they think the virus only makes the N-terminal part. So the capsid protein is not actually a nuclease. It's got mutations in the gene encoding the active site of the nuclease. But what they think is that the N-terminus of the capsid protein is positively charged. Now, this capsid protein actually binds the DNA of the virus. It's a DNA binding protein.
Right.
And they think the N-terminus of the capsid protein is involved in binding to the DNA. In the Cas4 protein, the N-terminus is covered up by the C-terminus of the protein. So it probably had to be split in order to become a capsid protein to expose that N-terminal DNA binding domain.
That's incredible.
And in fact, they looked in some other archaeal viruses, and some of those have Cas-like proteins encoded in their genomes, which have nuclease activity still. So this was probably captured at some point a long time ago. It was a nuclease that maybe participated in genome replication, and then it got split. The nuclease wasn't activated, and it evolved into a capsid protein.
Wow.
I just think this is so cool.
This is really, really neat. And I mean, how badass is that? There's this antiviral system in bacteria, and a virus came and stole a piece of it to build its capsid.
So they have a name for this. It's called exaptation.
Exaptation, yes.
It's when the function of a protein completely changed. So this is the first. We have had some examples where where proteins are clearly evolved from some other protein, but this is the first time when a capsid protein comes from something that's totally different, right? A nuclease becoming a capsid protein. I love it.
The two comments. The term exaptation is a pretty well-established term in evolutionary biology, I'm pretty sure. And what I thought was cool is that now we have the explanation of one capsid protein, and there are thousands that we don't really understand.
Well, yeah. But this at least points the way that this is the first evidence that this could happen this way. I think the whole paper is bioinformatics. That's the neat thing about it. So
there's one last point, and that is that in the title and the abstract and everything, they call it nucleocapsid, and then they call it capsid. And I've gone around with some students in the past about the difference between a nucleocapsid protein and a capsid protein. And Vincent, you might be in the position to adjudicate.
So a nucleocapsid is present as a substructure of a virus particle. So the influenza virus nucleocapsid is enveloped, has an envelope around it. Adenovirus is a capsid because that's the virus particle. Poliovirus, it's a capsid. If adenovirus or poliovirus had an envelope, then the capsid protein nucleic acid complex would be a nucleocapsid.
Got it.
That's really what it is. Now, this virus, it's an enveloped virus. So that's why they called it a nucleocapsid.
Okay, but then later they call it capsid.
Yeah, they're not consistent, yeah.
Okay. By the way, do all genomes of viruses associate on the inside of their capsid or nucleocapsid in chemical bonding?
Not chemical bonding, no. Well, usually non-covalent.
I didn't say it wasn't covalent or not. Just there's some attraction between the nucleic acid.
How many types of genomes are there?
7.
Thanks.
So is that a yes answer?
Do they
none of them are
in
just
some way?
None of them just float around inside the capsid.
Well, it hasn't been proven for all viruses. There are some cases where there are clear non-covalent interactions between nucleic acid
you're
and
working
capsid
on
proteins, but not for everyone has that been demonstrated. But
just wondered.
For the assumption is for packaging, you need to have some kind of interaction and so forth.
So these are highly selected chemical structures that have no homology to anything else in nature. That's remarkable.
Capsids.
That's remarkable.
Yeah. It is remarkable. It's really a cool story. So that's
crappy.
You're still there.
Awful.
Did you?
Still awful?
Yeah.
Yep.
Well, not personally, but your connection is.
Need to gargle.
Did you hear that, Rich Condit?
Yes, I heard the snippet.
Good. All right.
But if I'm really lousy, I'll just shut up here.
No, we're going to go to picks now, and we'd like to hear your pick.
Okay.
All right. Why don't you tell us, Rich, what is your pick?
My pick is a photographer I found named Ben Cooper. His site is launchphotography.com. He's a professional photographer that grew up on what we call here the Space Coast, so in the vicinity of Cape Kennedy.
Yep.
And he sort of has taken a gazillion pictures of launches of all sorts of vessels. And I think probably endeared himself to NASA at the same time and became actually employed by NASA as their launch photographer. And he's got a bunch of other stuff as well, because he's got travel photography from his travels and astronomy and et cetera. There's a really good link here called Popular that has some of his favorite and most popular pictures in it. And I just thought it was really nice photos of both nature and technology.
Yeah, he's got ballparks as a cool one if you're into baseball.
Look at this interior of the shuttle.
I was just going to I went to this too.
Oh my gosh, the control panel. I want that.
Yeah.
I'm working on that here in the TWiV studio, but I don't have anything like this. This is awesome. And so many wires in the walls. No wonder these things break. I'm still amazed that this and airplanes work. Oh, look, somebody autographed the walls. What is that all about? They're all their signatures.
They signed the inside of the shuttle.
These are all the people who worked on it, probably.
Yes.
Probably adds 10 pounds to the weight. Wow, this is awesome. He's got other cool stuff, too, of launches and stuff. Neat. Very good, Dr. Condit. Thank you.
There you go.
Alan,
sorry I'm fuzzy.
It's okay. No problem.
Great.
No, it gives you kind of a talking from Mars.
I was just going to say, like Pluto.
Have I been like this the whole show?
No, it just started happening.
In the beginning, you were better, but you should work on getting a wire because it'll be generally better.
All right.
And I don't mean a wiretapping kind of wire. But Alan Dove, what do you have for us?
I have a group based in the UK called Antibiotic Action. This is a nonprofit group that some people formed apparently to increase awareness of one of the great threats to global health, which is that our antibiotics are stopping working, as I'm sure you're all quite aware. And they are it's a nice site, and I think they've got a good idea that this is something that people ought to be a little more up in arms about because it could be fixed.
I saw an article the other day which said that physicians who try to be responsible with antibiotics are rated more poorly by their patients than others who give out antibiotics liberally. Yep. It's really too bad.
But the simpler problem to fix would be agricultural use.
Food. That's right.
No kidding.
That's the majority by weight of antibiotic use on the
Yes,
planet.
and we're pouring just huge quantities of these compounds into the environment for farming, and it's a horrible idea, and we now have the data to say that yes, that is causing resistance, like we're surprised, and that really needs to stop. I mean, there are entrenched interests that are combating that, but yeah. So anyway, this group is
not like gun control in the U.S.
Yeah, not to touch a red button issue or anything.
Kathy, what do you have for us?
I picked something. I can't remember how I found it, but it's a journal article and a video. The journal article is in AIDS Research and Human Retroviruses. It's by Marshall Bushman, Laurie Zimmerman, and Frederick Bushman. And it's called HIV Replication in Lego Mosaic.
Yes.
And the idea, the the purpose of this was to interest children and adolescents in learning more about HIV. So they created this HIV replication cycle in Lego Mosaic. So it's a stop motion animation, and it's pretty cool.
It is.
That's great. It's in the like you made a bunch of Lego videos, Alan, right? Oh, no, they weren't Legos, were they?
They were Legos.
Yeah.
I made a few of them.
Yeah, that's right.
Where they were figures, whereas this is a Lego board kind of drawing.
Yeah, very
thing.
cool. It's really neat. I like it a lot. And you don't know how you found this, right?
Can't remember.
Yeah.
It wasn't even that long ago.
Oh, the internet.
The internet. Dickson, what do you have?
Well, a dear friend of mine, Bill Campbell, just gave his Nobel Prize speech, and so I've picked that for my pick of the week. And it's a great follow-up to Alan's lament of the lack of development for antibiotics, because he has a plug at the end on why we're not discovering antibiotics at the rate we used to, and why we should go back to that method, because that's what led to the discovery of ivermectin, which is why he got the Nobel Prize.
Besides discovering more, we should probably take better care of the ones we have.
Tell me all about that, too.
Yes.
Yeah, that's exactly right.
All right, that leaves me, and I have a pick. It is a movie. I rarely pick movies, but I really enjoyed this one. It's called Ex Machina. Has anyone seen it?
Not yet.
No.
What's with you guys? What is with you guys? This
I don't
is
get
so
out
cool.
much.
No, you don't have to go out anywhere. I don't go to theaters either.
This is Netflix, right?
No, you have many streaming services. Anyway, this is a story about an android. You know, a guy builds an android. Actually, he builds a bunch of them, and they get better and better. And he throws out the ones he doesn't like. He kills them. And then this one realizes that he does that, and she plans to get out. So she has acquired sentience, basically. So I think this is interesting, because someday we will have robots that, you know, do human things, and maybe they will acquire sentience, you know, if we make them well enough. And that is interesting in itself. You know, you can imagine a whole slew of these robots rebelling and getting rid of the humans. Of course, when they're bad.
Old theme.
It's a very old theme, but it's fascinating to think about. And this movie really illustrates it in a wonderful way.
Okay.
You know, it's really good. But the reason I wanted to bring it up is together with the recent call by scientists, including David Baltimore and Paul Berg, to have a moratorium on edits to the human genome. All right, which we've talked about before. This is a New York Times article I'll link to. Scientists seek moratorium on edits to the human genome that could be inherited. Okay, we're using CRISPR, for example. And I was just thinking, you know, we don't want to edit people, but we're going to create people in these androids, but we're not regulating that in any way, as far as I can tell. And I wondered why. It's interesting to think about, right? Because you're going to create a robot that's going to get human. I think that's just as problematic as editing people and altering them and perhaps messing them up, you know, or making them want to. You can imagine editing a gene and all of a sudden the person goes crazy and kills everyone, right? That sort of thing. So it's an interesting contrast between the two stories that just, when I saw the Baltimore article in time, David Baltimore's on that article in the Times, I just thought, that's an interesting contrast between the two. You guys understand what I'm talking about?
No, I don't have a clue.
Hey, Carolyn, you still there?
Oh, I'm still here.
You enjoy this or you hate it?
I am enjoying it. Listening along and not having to contribute is nice.
Got so quiet. Do you want to make a pick? No.
Do I want to make a pick? I can't do that on the spot.
Yeah, you can. I know you could do it. What have you read lately that's really cool?
What papers have I read lately?
Or seen or done or...
I liked, and you know, this is a plug for a colleague of mine, although this is a paper that I really liked recently. But there was a nice paper by John Williams, who's our new chair of pediatric ID here at Pitt, on human metapneumovirus entry that was very good in PLOS pathogens. I liked that very much.
Okay, that's a pick. No problem.
Yep.
Metapneumo. So you have new people coming to Pitt, huh?
Tons of new people. Perhaps you've heard that Terry Dermody is actually going to be joining us at the University of Pittsburgh as our new chair of pediatrics, and we are thrilled and super excited.
So, what a nice guy.
Britt Glounsinger was here yesterday, and she told us.
Yep. He was one of my students, by the way.
Terry Dermody, yeah. We did a TWiV with him, the A-Train with Terry Dermody. That's very exciting, yeah.
Great stuff.
Oh, we're really excited. Yeah.
and it's in Pittsburgh, of all places.
Lucky you guys.
Who would know? Who would have thought?
Pittsburgh's a lovely city now.
We have good wine, and I think that was probably something that had to be factored into Terry's recruitment here, I guess.
What?
Your football team's doing well right now. Your baseball team did great. Come on.
I've never been to Pittsburgh.
Really?
What? Well, you're coming in January, so you'll get to see it.
Pittsburgh's.
All right, we have 2 listener picks. One is from Tom. This study has a beautiful graphic that matches animals with their known microbes and draws connections between species with similar microbial crews. Science News described it as a social network of species that resembles a vibrant tangle of yarn. And this was in Nature. It's a pretty cool figure. All right, so you guys are not going to be able to see it, your listeners, but it's a very cool picture. And then Trudy writes, Dear TWiVers, I would like to suggest a pick of the week, and I really hope it hasn't been picked yet. I recently finished reading Madame Curie, a biography of Marie Curie, written by one of her daughters. I first heard about this book when Vincent mentioned it on a very early TWiV. I immediately made a mental note to read it. However, it has taken me a while to get around to it. Somehow this woman really did it all. Not only did she excel in her career, but at the same time she also managed to be a loving wife to her husband Pierre and a loving and involved mother to her 2 girls. At least that's how her daughter Eve describes her. The book describes her struggle with fame and fortune and how she pretty much gave away all her money. She used a lot of it for her research, but also donated quite a bit to create radiological cars, which basically provided x-rays on wheels for those injured during the war. She also installed 200 radiological rooms in hospitals in Paris, a novelty at the time. She continuously shied away from fame and once mentioned to a journalist that in science we must be interested in things, not in persons. I disagree with her, and it seems that so does Arturo Casadevall. Coincidentally, I recently ran across one of his papers in the journal Infection and Immunity. This paper discusses the importance of familiarizing oneself with the history of science. She gives a link to that. When we think about people like Marie and Pierre Curie, it's easy to assume that their hypotheses were always correct and that their results always materialized miraculously and effortlessly. I think it's important to be reminded that this is not the case. Casadevall does a great job of it. Quote, major events in the history of science are widely known and well documented, although the intellectual and experimental struggle required for discovery may not be as well appreciated. As any working scientist knows, the process of scientific discovery is messy and often involves dead ends, chance, and being in the right place at the right time. Moreover, scientists who are concerned with only the facts and not the process miss out on the rich human drama of perseverance, serendipity, inventiveness, and conflict that characterizes the history of science. It is often such details that are most interesting to a non-specialist, which in turn facilitates teaching and the engagement of the general public with science. The omission of the history of discovery from scientific papers may thus serve to perpetuate the barrier between scientists and the public whom they serve and depend on for support. Well, there you have it. I have nothing else to add. All the best and keep on TWiVing. Trudy.
I have to concur on the Madame Curie. It's probably the first biography of a scientist that I read. It was a copy that my father had, and I still have that copy. And I think I was junior high age when I read it. It was really impressed me.
Cool. By the way, just skipping back to the previous listener pick, the nature figure is freely accessible.
Nice. It's a cool figure, isn't it?
It is a very cool figure, yes.
It's really well done. Thank you. Very good. Excellent. Yeah, we need to know the history of science because it doesn't all work, It's not all smooth. And people may think that, but it's not the case. But here on TWiV, we give people the real deal, right?
Yep.
We talk about controversies and disagreements and so forth. And it's all on TWiV, and you can't get anything like it anywhere else. And if you've made it this far, congratulations. It's 2 hours.
Yes.
Thank you very much. This is TWiV number 367. You can find it at twiv.tv and also over at iTunes. And we like to get your questions and comments. Please send them to twiv at twiv.tv. I would like to thank our special guests, Coyne Drummond, who left us already, and Carolyn Coyne. Thank you, Carolyn.
Thanks for having me.
It was a pleasure. You come back sometime?
Sure, it was great fun.
And you're very clear.
Good.
We can. The mic works great. Thank you to M-Sphere for doing that. Mm-hmm. Dickson Despommier is at verticalfarm.com. Thank you, Dickson.
You're welcome, Vincent.
Did you enjoy today's episode?
A wonderful time.
Oh, yes. Kathy Spindler is at the University of Michigan, which is in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Alan Dove is at turbidplaque.com, and you can also find him on Twitter. Thank you, Alan.
Thanks. It's always a pleasure.
And the fuzzy Rich Condit is in Gainesville, Florida. Thank you, Rich.
That's the title of the show.
How do you spell it?
Always a good time.
How do you spell that, Dickson?
You have to ask Richard.
Ooh. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
Am I still
crummy? You're really crummy. Can you go somewhere else? Get closer to the
router.
To the router.
Oh man.
Put some tinfoil on your head. That should take care of it.
Stand on one foot.
Oh, this is wonderful. That's good.